So good afternoon again for all delegates and participants. So this is the second webinar of the ITTO-TIC and variable species plantation or the project phase 2 co-funded by ITTO and the Ministry of the Food and Agriculture from Germany. Yeah. So if you notice the logo carry, so you see that we have the new logo. This one, this is the Ministry of Forestry from Indonesia. So previously that the Ministry of the Environment and Forestry now it have been reorganized to separate Ministry of Environment and Forestry. So we have Ministry of Forestry with us. So for those who are just joined today webinar, so I would like to briefly introduce that this project is funded by Ministry of Food and Agriculture from Germany. So we have approximately to your presentation from November 2023 to the end of December 2076. So we have 5 countries in Asia Pacific participating this country, including Cambodia, Thailand, Vietnam, India, and Indonesia. And we also have Togo in West Africa to join this project. So for today's webinar, so the title is Quality Planting Material for the Premium Teak. So we have the 2 prominent speakers from India, Dr. Yasodha and Dr. Suwan from Thailand to deliver a presentation today. So before we start the webinar today, so I would like to invite Jennifer Conje, the Director of Forest Management Division from ITTO, to give the open remark on behalf of the project. So Jennifer, please.
Thank you very much, Dr. Youngyard. As he mentioned, this webinar series is part of the second phase of the ITTO flagship project promoting quality timber production in smallholder and community-based teak and other valuable species plantations in the tropics. As Dr. Youngyard noted, it is supported by the German government and has been under implementation since 2023 in Cambodia, India, Indonesia, Thailand, Togo, and Vietnam. Now, the theme of this second webinar is quality planting materials for premium teak production. I extend my thanks to our invited presenters, Dr. Yasodha Ramasamy from ICFRE-Institute of Forest Genetics and Tree Breeding in India, and Dr. Suwan Thangmitcharoen from the Royal Forest Department of Thailand for sharing their expertise and insights during this second webinar. The topic of today's webinar is very important. It's the start of the supply chain. Without quality seeds or seedlings and ensuring survival, quality, growth, and resiliency during the teak tree's formative stages, one cannot progress to the next step in the supply chain. We have learned from the first webinar held on February 14th, the importance of sustainable tropical forest, where 1.6 billion people depend on it for sustenance and livelihoods. The teak tree is widely cultivated by smallholders in combination with other agricultural crops or alone as an alternative livelihood option. Teak-based small-scale production systems enable farmers to diversify farm production, support food security, generate income, and reduce financial risk. They serve, in a nutshell, as a farmer's living savings bank to meet current and future financial needs. These webinars are created to provide a collaborative learning platform for smallholders, forestry authorities, and academic partners to advance knowledge and innovation on a diverse set of issues across the supply chain for teak and other high-value timber species. So I encourage you to be active in your engagement during the Q&A period. And again, thank you to our speakers. And I look forward to the fruitful discussion. Thank you, Dr. Youngyard.
Okay, thank you very much, Jennifer, for your kind opening remarks. So without further ado, I would like to start with the first speaker. So Dr. Yasodha Ramasamy is the scientist from the Division of Plant and Biotechnology from ICFRE-Institute of Genetics and Tree Breeding in India. So she is the prominent scientist in terms of genetics and DNA. So recently, she published a book on the teak genome, as you can see on the skin. This very interesting book that you can find more further information, and if you are interested. So now I would like to invite Dr. Yasodha to give a talk on teak genomic resources. Okay, just a moment. I will share her PowerPoint.
Thank you, Dr. Youngyard. I hope I am audible to everyone. Professor Youngyard, you need to unmute. I think you are trying to say something.
Okay, you can go ahead, Dr. Yasodha.
Yeah, thank you very much. Good afternoon, good morning, good evening. I think different parts of the world are connected today. Thanks for the opportunity given to me. Miss Jennifer Conje, Director, Forest Management Division, ITTO, Dr. Ma, Dr. Tetra from ITTO, Professor Youngyard, and co-speaker Dr. Suvan, and many eminent scientists and researchers available online. The topic I have chosen is on genomics of teak. When Professor Youngyard approached me for a lecture in this second webinar arranged by ITTO, I felt that this is the right point for me to talk something on genomics, because genomics in other fields like say humans or animals or agricultural crops, it already started yielding its fruits. So forestry is a bit behind. So we thought that why not we tag this kind of discussion in this webinar, so that we are encouraged to take up such advanced work. If we see forestry as it is, one species, Populus trichocarpa, was sequenced somewhere in 2006, and it was published. Then after that, most of the temperate tree crops were taken up, and very little work on tropical trees. So at that time, we approached, like from ICFRE side, we approached Department of Biotechnology in Government of India, and looking for a support. Why not we take up some advanced research in forest trees? So we, IFGTB, along with the Kerala Forest Research Institute, we have taken up a project for whole genome sequencing. From then onwards, the Department of Biotechnology is continuously supporting us, and we are trying to add more research in this field. Coming to the current slide, which is narrating about the genetic variability available in teak. Of course, all of us know about the importance of teak and its multiple uses, everything. It is for generations we are using teak. Coming to the genetics part of it, It's like any other tropical tree. It is highly outcrossing one and harbors much of heterozygosity in it. And specifically for teak, distinct provenance features they have. For example, teak from Andhra Pradesh is something unique. It has got certain features unique. Likewise, teak from Kerala. Kerala also got multiple provenances. Like that, the entire distribution range, not only India, in Myanmar, Thailand, and Laos, quite a good amount of diversity is available, and these diversity should be utilized for tree improvement purposes. And clonal forestry is the one which brought in a lot of improvement in tree species, particularly species like a previous slide, Dr. Yongyut, please. Populus or eucalyptus, these species have become more popular because not only because of their short rotation, because of clonal forestry. Clonal forestry is the one where you can get both additive and non-additive genetic features quickly, and we can transfer the results to the field. So when we are targeting teak for short rotation, definitely the clonal propagation part is important, and the species as it is, teak is amenable for clonal propagation. May I get the previous slide, please? Professor Yongyut, may I get the previous slide? Yeah. Previous one. I was just narrating about the genetic variability existing in teak.
Okay, this slide?
No, the first one, what I was talking about. There I have given something about the genomic features. Yeah, this is the one. Thank you. And teak has some higher influence of environment. Wherever it grows, it is the environment plays a major role, like in the growth features as well as morphology. It has the environment plays a major role. And coming to the adaptation part, it is highly adaptable species also. There are certain research already.
Dr. Yasodha? Dr. Yasodha?
Maybe network failure.
Dr. Yasodha, I lost your connection. Dr. Tetra, do you hear me?
Yes, I hear you.
Okay, Dr. Yasodha lost connection.
I think we can proceed with Dr. Swan and then go back to Dr. Yasodha after that.
Okay, good. Dr. Swan, are you ready?
Yes, I'm ready.
Okay, good. Okay, go ahead, Dr. Suwan.
Okay, may I start now? Okay, good afternoon, everyone. Very good opportunity for me to share the information on the teak genetic in Greater Mekong subregion. This is my I adjust the title because due to the time limitation. So I think it's better just to focus on the teak genetic in Greater Mekong subregion, which is narrowed down from Dr. Yasodha. No? I talk about the genetic in Greater Mekong sub-region. Okay.
I'm sorry. Maybe we have to inform Dr. Yasodha.
Dr. Yasodha, due to we lost your connection, so may I ask your permission to start with Dr. Suwan, and we'll come back to you later. Okay?
Okay, right.
Okay, thank you. Dr. Suwan, go ahead, please.
Okay. So sorry, Dr. Yasodha. Okay, may I put?
Okay, fine. Carry on.
Yes, okay, may I proceed now? So I will talk about this topic. First, I will talk about the genetic resources. Of course, when we talk about the genetic, we cannot avoid with the conservation. So I will mention about the in situ and ex situ conservation activity. And then the three improvement in our region. And then I will tell you about the recent research project, which is the deal with the molecular biotechnology, especially the breeding, breeding technique, and then summary. First of all, I would like to say thank to the ITTO project. Much of information I present here I gather from the face-to-face meeting or seminar, and including field trip where we go to Myanmar, Laos, not India yet, no. And also we have a chance to visit Vietnam because I'm one of the chair of the technical committee, thanks to the ITTO project as well. So I just give you hints of the each country where we know that this is the native country for teak distribution. And in general, you can see that only Myanmar now pursue for the natural forest logging, but after we have no logging in the natural forest. And most of the forest where we have teak is deciduous forest. And let's have an example for conservation of teak resource in Laos. I would say that not much information for Laos, and the only most of the information we have in Laos is we call it seed source, where the largest occur in the Chayaburi province. And in Myanmar also present that this is the largest natural teak bearing forest area in the world. It claimed like that. But I think most of the natural distribution in the mixed deciduous forest and deciduous littoral carp forest. Let's talk about Thailand. This is the natural teak distribution in Thailand. We have in both protect area and outside protect area where we call natural reserve forest, and much of the area in the occur in the protect area. This is what we call in situ conservation. Most of the in situ conservation distribute in the national park and wildlife sanctuary. Also, we have we call biosphere reserve in Lampang province in the north. This is special area for the conservation. We also have the new finding of the high elevation of teak distribution. In the past, we only think that the elevation is only up to 600 meters above sea level, but just maybe later we found that we have high elevation for teak as well, 1,200 meters above sea level in Mekong province in the north of Thailand, far north of Thailand, close to Myanmar. Well, then other than in situ, when we talk about the tree improvement program, you all know that we make it as the ex situ gene conservation. We have gene bank, seed orchard, seed production area, provenance time, progeny and clonal test. This is the sample principle of the tree ex situ conservation, especially for tree improvement process. This is just to focus again that when we talk about the tree improvement process, it can be structured like this. And the most important thing you can see that input technology, where we have to make several kind of the technique. It can be reproductive biology, vegetative propagation, and also DNA molecular. We have this kind of information long time ago, where we try to promote to use the better germplasm, improve genetic material, because we know that we can get a high grain from the tick improvement program. And this is just to give you the idea that in general, now we can say that we are at the second era, or what we already learned that we call it conventional tree breeding, where we have few try like Conan test, provenance try, or progeny test. But we are moving toward the third era, try to link breeding and biotechnology together. This is the example of teak seed source in Laos, and or we call we can say in situ conservation. This is example of the head orchard in Myanmar. They also have head orchard as well. I hope it's still in the good situation now. We visit here maybe around 3 years ago, I think. This is the information of the clonal seed orchard in Myanmar. They have several plots of the clonal seed orchard also. And very good that in Myanmar, they also have study on the geographic variation in Myanmar. It can be divided into 4 clusters, 4 groups of population. This is a very good information where we go the next for further step for the tree improvement program, and they can define that some area could be considered as the highest priority for conservation because of the high variation of those areas. And for the genetic diversity, it's considered as high because the expected heterozygosity. I think Dr. Yasodha will also mention about this as well. 0.65, this is considered as high heterozygosity. In Thailand, we also try to make a grouping of the population as well. And nowadays we use this as the database when we make breeding, or we try to make hybrid vigor. So we try to, when we make a controlled pollination, we have to use the material from different population. We also have the plastic selection, almost 600 plastic already select, most of them in the north, and also some part of the outside of the natural distribution in the northeast. Also in the plantation, we also select as the plastic as well. And some activity test, progeny try, and the tissue culture technique apply. This is the example of the improved seed source, which is composed of the seed orchard and seed production area in several countries. And we go further to find the good clone, where the criteria, the main criteria is the good growth and the hardwood proportion, high hardwood proportion, and we get some at least 10 cone that we find very good performance on this aspect. We also try to put the better germplasm, improve genetic material to the tree farmer. And this is I try to compile status of the tree improvement in Mekong region, especially in Laos and Myanmar. So I think for Laos and Myanmar, still need to do some protein test or clonal
มันลื่น
test
มันลื่น
if possible.
คนน่าจะนั่งตัวนี้ดีกว่าเพื่อให้เขาให้
Most of them already apply no
นักตัวนั้นก็ตัวนี้ลื่น
three prominent try in
มันไม่เห็น
those
รูป
two countries, Laos and Myanmar. And this table you can see that Laos or Myanmar already have the seed orchard and the seed production area or even seed orchard. But for Laos still has no head orchard. But interesting to see seed production area in Myanmar is high, as you can see, more than 3,000 hectares. And for Thailand, we have a big area of seed orchard, almost 1,000 hectares. Let's go to some study, what we call molecular biotechnology. It has been working for a few years already. We cover something like DNA fingerprint to make. We try to make database of the clone available that all the in or the gene bank. We already have it, and also we produce the new breed for control pollination and investigate cultivation technique for tissue culture and establish database for supporting of the genetic improvement. This is the way we do control pollination. Yeah. Make a full zip progeny. This is the example of the new breed that we have. We put the number nowadays. We have 10 new breed, and it's become more and more because we still make a new breed in some research project. And also we have ongoing research on molecular genetics for teak breeding. We expect to get genotyping of all clones that we have, as already mentioned, around more than 500 clones, and focus on the important trait for the timber production, genetic marker used for further selection. And the important thing is we try to develop the model to estimate future trait for the seedling. We don't need to, it means we don't need to wait for the testing until maturity, as we know that for the conventional breeding program, we have to wait for maturity to measure and to see the form of the tree that we are testing. Meaning that the model can help to accelerate the teak breeding program. This is our team making the molecular breeding strategy, where we make it from colon test and progeny try plot. Okay, I mentioned a bit. We have the another organization that come to work together with us is the JERCAS, the Japanese corporation, JERCAS. I think some of you may familiar as well. We got the material. This is the colon test at Kanchanaburi province. We measure the tree and also the hardwood proportion as well. This is the progeny try in Donglan in the northeast of Thailand, where we have the Funsip progeny. And this is the model that we expect to get the genomic prediction model. We try to reduce the time for the conventional tree breeding, try to set up the model, and we just estimate from the seedling. Then we can get the trait that we would like to get in the future. I just give you some interesting information from just very new. Actually, this information has not been published yet, but I think that it is interesting to see this kind of information. So I will draw this message to tell you first. Now, we found that the hardwood proportion should not be well correlated with the diameter. It means that if we even have a bigger diameter, but doesn't mean that those big diameter has a big hardwood as well. In other words, we can say that the stem and hardwood do not show high heritability. But another way we found that high and sapwood show higher heritability. Okay, let's come to the end. So in general, I would say that for in situ and ex situ conservation already apply in Greater Mekong region. And genetic activity among these countries vary on the breeding program. Some countries already have progeny or colonies or molecular breeding, but some countries may have to set up this kind of breeding program. And interesting to see the high genetic diversity in Myanmar and in Thailand. And once again, the high and sapwood, they show higher heritability. Or another word, I would say that the hardwood proportion of hardwood depth may be depend on some environment as well, not just the genetic power. Well, let's think ahead about the challenge work or future work. For long-term breeding program, I think we should be promoted in Greater Mekong region, and the tree breeding program, if we can make cooperation, would be better to get the more effective of the result of the breeding program. And biotechnology in conjunction with the intensive breeding program need to be developed, because we know that the biotechnology has become more and more important and could be make the tree improvement program more effective. And for the access of the better germplasm, I think this aspect, many countries has some limitation. We try to promote the good genetic material, better germplasm to the tree farmer, but still not very good. No? There is some limitation of this aspect. And of course, sharing status knowledge and technology among Greater Mekong region countries is important. So we know each other what we are doing or what we should go together in the future. And cooperation among the regional agency with the cooperation of the very big agency, you know, ITTO or TICNET, UFRO, need to be strengthened. And lastly, the young planted teak need to find innovative to the market. This aspect, I put this because now we also have the project research that we are trying to use the young age or young size of the teak wood, try to make like engineer wood. It could be CLT or glulam to make some kind of wood construction or building, housing, or something like that. Okay, this is what I would like to mention. Thank you very much. And if you have any suggestion or sharing, very welcome.
Okay, thank you very much. เอาสไลด์ลงเลยฮะ Thank you very much, Dr. Suwan, for your informative presentation. So I would like to make a short summary. So Dr. Suwan provide the overview of the natural teak forest in Thailand, India, Myanmar, and Laos, and also the overview of the study and research in the 4 countries. So he emphasized in Thailand, in particular, the teak improvement that can be subdivided into era of the study, starting with the botanical garden seed orchard at the beginning with the financial and technical support from Darida since 1965. And then the second era emphasis on the connotates for Nochinis for the 568 mother tree. Now we are in the third era that emphasis on the DNA and molecular technology. So that's the overview of his presentation. So I open the floor for 2 immediate questions, and for the general question or discussion we can discuss after Dr. Yasodha presentation. So anyone would like to ask a question? So let me allow 2 questions. Please raise your hand. Or you can ask a question. Any question from the floor right now?
Yes, I have a question.
Yes, please identify yourself and organization and ask your question. Go ahead, please.
This is Nuruddin from ITTO. This is Nuruddin from ITTO. Hello, can you hear me?
Yes, I can hear you. Please.
For some reason, my video is upside down. I don't know why. So, but this is Nuruddin from ITTO. Yeah, thank you very much, Dr. Suwan, for that wonderful presentation. For me, it brings me back to my graduate studies days, long time ago. And I have learned a lot, especially looking at the advances that you have already presented with regard to breeding in teak. Yeah. In your presentation, you mentioned that I also saw, that in Dr. Yasodha's presentation before she went offline, that teak is actually highly outcrossing with very high genetic diversity, which is good news. But in your presentation, you also mentioned that there is high correlation between height and sapwood. High correlation, high heritability between height. High heritability between height. Okay. However, hello? However, heritability for diameter and hardwood is rather low. It's rather low. And I think in trade, we would rather want to see a very high heritability that it can pass on to a much higher, bigger diameter that also has a very high, hardwood. That would also imply some high level of density. I mean, that is what I know. I mean, from going back to my prior studies when I was a student. So how do we try to now use technology itself to bridge the gap between selection for higher breed with very broad hardwood and also maintaining high level of genetic variability?
Okay, thank you very much.
Yeah.
Dr. Suwan, please.
Sorry, can I get your question again?
Okay,
You mean how to read?
Maybe I can repeat a question again. So in your presentation, you show that there is very low correlation between the height and the percentage of hardwood.
Yeah.
So the question is there any innovative technology or study that we can enhance the relationship between the height and the percentage of hardwood? Because hardwood is very important. Because
Exactly.
if we have more
Yeah.
hardwood, more density, the price will be much
Exactly.
higher. Yeah.
You got it right, Professor Young Youth.
Thank you. Thank you. Dr. Suwan, please.
Thank you for the clearer message. Well, if you think about the technology now, is I think I would say I cannot think it of now, but as I mentioned a bit about the hardwood proportion is most of, so for this information that we found, one thing that we can answer is the hardwood is may or most of them rely on the environment. So what we are trying to do, maybe we can do something with the environment. Actually, we've been thinking about some technique like girdling to make I'll cut some branches to activate the occurrence of the hardwood. To how to say it, I like try to make the tree some straight, put some straight to the tree. This is the one thing that we should be possible. We think that it should be possible in practice. I give you some example. Maybe it can be seen clearer. If you saw example, if you have some big tree and growing in general way, but if you try to cut a big branch like that, many branches and leave it, so sometimes the hardwood seem to be bigger than the one that they didn't cut. This is very simple technique that could be possible, but I mean, other than if you think about scientific way, we still has no information on this. Or just try to make another way is try to find the plastic where we have more hardwood, but still maybe not really good. How to say, good try, because the low heritability.
Okay, thank you very much.
For now, I can say only this. No, thank you for your
thank you.
question.
So the answer is we
Thank you,
don't
Dr.
have
Suwan. Thank you very much.
Yeah. So the answer is we don't have the scientific evidence to show Okay, but right now he said that we use the manipulate environment and silviculture techniques. So I will provide you for further information for this project for the Teak phase 2. We have the chrono test. So from 568 mother tree in Thailand, we already identified 75 champions, which have the more proportion of the hardwood. So the demonstration part and experiment still going on in two areas, one in Chiang Mai and the other one in Kanchanaburi province. So one more question before we proceed to Dr. Yasodha.
Yes, Prof. Yongyut.
Yes, please identify yourself. Yeah.
Yeah, I'm Rina from the Ministry of Forestry, Republic of Indonesia.
Okay,
Maybe just a
Dr.
little
Rina.
bit, regarding what is explained by Dr. Tuan about the management of the teak plantation in the preservation area, preserve area, conservation area. I mean, it is a very different condition with Indonesia, and it is very interesting. I mean, like, can you just how to say, explain a little bit more how you engage the community, and then what is the challenge, because we cannot harvest anything from the protection forest. So it's totally different condition between Indonesia and Thailand. So it is a very, how to say, interesting actually to, to get information and about that one. Thank you.
พี่ฟังไม่ทันฟังได้ไหมพี่จัด
So I think if I can summarize, because it's in the tropic area. So the question is, Indonesia do not have the natural teak forest, but they have only the plantation. It's more than 400 or 500 years ago. So Indonesia do have the what we call the good genetic resources. So in case of the Indonesia, any of suggestion from Dr. Suwan?
I think in the past we've been when we have the teak improvement program, we one thing that we should try to make it is the germplasm collection. We make germplasm collection from teak. Try to cover the teak distribution as much as possible. And I think it's come to, it may rely or go align with my summary or my suggestion that to make more effective tree improvement program that you mentioned, we should try to make it would be international project, make it together to make the regional tree improvement program so that we can compile the germplasm of teak in at least a native country where the teak grow naturally. So in this way, in the past, we already learned that if we try to cover the natural distribution of the tree improvement program of any species, we found that it would be very effective. I mean, the production of the tree improvement or the better germplasm will be more effective. Can you get me?
Yeah. Okay, thank you very much. Yeah.
Yeah, maybe a little bit of question, Prof. Dr. Swan. How is the comparison of the productivity between the production forest and also the protected forest or conservation forest?
Natural and plantation?
Yeah, because the natural is in the protected or in the conservation area, right? Not in the production forest area. So it might be slightly or maybe has a significant difference the productivity of the teak plantation itself.
Do you have any evidence about the growth performance between the natural teak forest and plantation, Dr. Suwan?
Well, I think in general, what we are doing now, we try to make a breeding program. So as I said, if we have effective breeding program, it will be more production. But if you remember my maybe third slide already mentioned the gain try, you will get more gain try from seed orchard or some vegetative propagation where we already make the improved genetic material. This is I think this is the evidence, good evidence. That's why we have to develop effective breeding program instead of waiting for national. For national, you can just go and just select the plastic only, but there is no develop of how to say, develop the genetic base. This is what the most important thing for the tree improvement program. We go step forward or try to make more productive by this technique now.
Okay, thank you very much. Yeah, now then we can proceed to Dr. Yasodha. But before Dr. Yasodha make presentation, may I ask your cooperation to mute your microphone? Okay, then let only Dr. Yasodha. Suchanat, can you share the PowerPoint of Dr. Yasodha?
Sorry, sir, I don't have presentation.
Okay, I can share. Just a moment. Okay, let me share Dr. Yasodha. Okay. Okay, do you see the slide? Dr. Yasodha, do you see your presentation? Dr. Yasodha, do you hear me? Dr. Yasodha. So anyone hear me?
Yes, I can hear you.
We can hear you, Dr. Young. Yes.
Okay, go ahead, Dr. Yasodha.
It seems she lost the network.
Dr. Yasodha, do you see your PowerPoint, your presentation? I already show on the screen.
Dr. Yasodha, I think you may be muted on your side. We cannot hear you on our end. Your microphone is still not working.
Dr. Yasodha.
So maybe Tetra, you can continue to work with Dr. Yasodha on the side, and then Dr. Youngert, maybe just take more questions from the audience in the meantime.
Okay. Yes. Dr. Yasodha, I think you mute your microphone. We do not hear you. I cannot hear you. Okay. So maybe we can take more questions from Dr. Suwan's presentation. So if anyone have more question about Dr. Suwan's presentation, please.
Professor, are you able to hear me now?
Yes. Okay. Okay, now it's okay.
Okay.
Then I will share your presentation. Just a moment.
Yeah, please.
Okay, go ahead.
Sorry, some interruption happened. Continuing my presentation, I was talking about the short rotation teak across the world, not only in the native regions, mainly in the introduced locations like Latin America, Africa, and certain part of Australia is cultivating teak on short rotation, and smallholder plantations are highly promoted also. And teak has a specific future, like in the initial 20 years, it shows a fast growth. So that is one benefit we reap in the short rotation plantations. Here, definitely we need to identify the individuals which can give better heartwood in this period of shorter rotation, say 20 to 30 years. We should be able to get a maximum of heartwood. That is the objective in many of the breeding programs. And teak aceties, we use them as a furniture or buildings like that. So carbon sequestration is also happening there. So it indirectly gives a solution for climate change problems. So because of these reasons, we are interested in introducing the genomic based tree improvement tools, so that the improved planting stock can be produced a bit early compared to conventional methods. And as it is the provenance genetics of teak, like at the international level, somewhere in 1930s and 1970s, there were two different time zones. They started the international provenance trials. In the year 1970, rather a bigger program, like 75 different provenances were planted in 50 different sites in 19 countries. Even now, some of the locations have these trials, and these trials form the source for the planting material. And initial results of initial 20-30-year-old results published by different researchers show that certain provenances from India and Laos perform better in the moister locations. And particularly the Indian provenance, Nilambur provenance, is doing better in the in many locations. It shows the highest stem straightness as well as good heartwood. So we need to really identify such provenances.
Dr. Yasodha, I'm sorry, can you speak louder? We cannot hear you.
Yes, Dr. Suwan told that here the age correlation of phenotypic traits are much higher. So this is actually helping us to identify the superior provenances. And as I told you earlier, that clonal propagation is the one tool which helps us to capture these characters fast, and we can take them to the plantations. Coming to the population genetics of teak, like many of the studies have shown that provenances are unique. And in this slide, we can see that this is the work done on the international provenance trials by the Denmark group. Dr. Hansen and others have published this. We can see the circles here. They show that they are the unique genetic clusters like that. In India, the southernmost, like Kerala, Karnataka, Tamil Nadu part is something unique with the highest genetic diversity existing there. Then in interior central Indian dry teak is having a special kind of genetic makeup. Likewise, in Myanmar also, there are multiple genetic clusters existing. When it is moving to Laos, at the end, the diversity is getting reduced like that. And next slide. And this slide gives the population structure, one by the international group, international provenances tested by Hansen et al. Another one study, Balakrishnan et al., where we have studied about 19 different provenances within the country, mainly southern part of the country. And both the results are almost matching, that these kind of population differentiation is something unique for teak. So each and every population is important for us for our future tree improvement programs. Next one. This study again shows that international provenance trials, how they are distinct. So I cannot say that these populations in India are unique or something like that. Every individual population present in these naturally occurring areas like Myanmar, Thailand, and Laos have their own unique genetic features. But if we see the genetic differentiation, that the FST values I have shown, the color intensity is high, that is much higher in the Indian and Myanmar populations. Next one. Yeah, this slide, of course, Dr. Suvan has also shown there are 4 different genetic clusters existing in Myanmar. They studied using chloroplast SSR as well as nucleome SSRs. And if we see that how genomics is going to help the tree improvement as it is, we have already seen the kind of genes existing in teak, and how many genes are controlling the hardwood traits. All those have been studied elaborately, and this information will help us to fast forward the existing breeding programs. And genome-assisted breeding has already proven that the generation time is getting reduced using these kind of advanced tools. Next one. The Brazilian group has also worked on the transcriptome resources as well as functional genomics part. Particularly the Professor Helen Carrier's group has worked on transcriptome of different tissues, and they could study the cytogenesis part, that is the part governing the hardwood formation, and as well as the climate change related traits like abiotic stress tolerance. So they are trying to put in more of information how the genes are linked with these traits. Next one. In India, we are trying to work on genomic selection part. This particular like this strategy, Dr. Suvan has also explained that using the large number of DNA markers, we can speed up the breeding. Conventional breeding program can be we can speed up the programs. Next one. We are currently working on genomic selection. This is again the Department of Biotechnology funded program. Here, University of Copenhagen is also involved. Dr. Eric is the principal investigator from the other side. And we use the technique called FTIR, that is Fourier transformed infrared spectroscopy, where the chemical signatures are studied across the clones, and we are trying to link them with the genetic information. And our main target is the hardwood content as well as the tree ring size. These are the parameters we are working. Of course, wood color also matters. So we are working on that. Coming to the international initiative as it is, of course, we know that under this Greater Mekong Subregion project, now India is also part. We are working towards the smallholder teak improvement and conservation of teak genetic resources and all. And India works with Denmark in the genomic selection part, so that we can develop the genomic tools that will be used for identification of individuals with high hardwood percentage. And next one. Currently, not only the region where teak is occurring naturally is the countries are working on tree improvement program. Other than that, Malaysia, Central and South America, and Africa all have the tree improvement programs in place. Next slide, please. I propose this if we have an international institute for teak, mainly focusing on conservation as well as breeding aspect, that will give us more results, and we can speed up the adoption of these protocols in many of the tropical regions where teak is growing. And definitely we have to have a coordinated teak gene pool establishment for natural populations as well as land races. We have still unexplored diversity in the economic characters as well as ecological characters that we need to work out. And this is a time we need to integrate high efficient breeding tools like genomic selection or CRISPR technology, all need to be integrated. And coming to the summary, like teak definitely demands a robust breeding plans at international level because many of the tropical countries are going for commercial production of short rotation teak. And teak genome studies offers powerful tools to accelerate conventional breeding and many other aspects where genomic tools can be used. And the wood industries, they are interested in short rotation teak, but they are also looking for the quality. So definitely we need to improve those quality traits like wood color and hardwood content, increase the extractive, that metabolites content, all those should be given priority so that the life of the short rotation teak is extended like the conventional one. And we also need to give the importance on pest tolerance of the clonal plantations and of course on climate change adaptation potential, all those need to be prioritized. That's all from my side. Thank you very much.
So thank you very much, Dr. Yasodha, for your presentation and summarize the key message from your book. So according to your presentation, can I conclude that the genomics technology can enhance the quantity of hardwood that Dr. Noruddin asked Dr. Suwan previously? Dr. Yasodha?
Yeah, genomic selection is a tool. We can use that tool for increasing or identifying the individual which has more hardwood content.
Okay. Can you talk close to microphone? Because your noise is quite low. Thank you. So are there other questions from the floor?
I do. I have a follow-up question for
Yes,
Dr.
Dr.
Yasodha.
Noruddin, please.
Yeah, thank you very much, Dr. Yasodha, for the insightful presentation. I have learned a lot from your presentation together with Dr. Suwan's presentation. Yeah. So you have mentioned that it is possible that we can use genomic breeding to be able to also enhance the hardwood of progenies that would definitely later grow up to be mature trees. However, I'm just wondering, wouldn't that also, number one, compromise the level of genetic variability? And if level of genetic variability is compromised, what happens to plantations, for example, in the advent of climate change, where we are now looking for three species to have very high plasticity, that is their ability to be able to adapt to climate change. With genomics, wouldn't that be compromised?
Like genomics as a tool or as a technique, it is not going to compromise the diversity or anything like that. Always the tree improvement, when we domesticate the genetic material, the diversity is coming down. That is natural. So we need to plan in such a way that we allow multiple clones for plantations. They always say that clonal mix should be there so that pest tolerance is also controlled. Likewise, climate change adaptation. So we need to balance out this. This is the duty of the breeder to give the diverse material also at the time productive also.
I have another follow-up question. Sorry, I'm taking time to ask questions. Yeah, I'm happy you mentioned that you need to have a mixture of different clones. I actually taught a plantation, teak plantation in Panama, and they had a plantation where a 14-year-old plantation was given between 35 to 39 centimeter diameter, which is quite good. I mean, excellent for teak. Yeah. So I asked them how many clones do they normally mix so that there is a level of balance in terms of variability, just in case of maybe insects or pests attacking plantation. And they told me that it is 11. I asked them, why did they say 11? Is that optimum? They were not able to tell me because they are just people in production. But I want you to tell me from scientific perspective, what is the optimum number of clones that you think one should use for plantation, especially commercial plantation, and also take into account climate change?
We need to work out that we cannot say arbitrarily this number is good or like that, and how those clones are related, all those things. We recommend generally 20 to 25 clones like that, but it is difficult on a large scale plantation propagating 20 or 25 different clones and taking into the plantation. So we have whatever maximum possible for handling. Wise decision need to be taken at that time, right, by the breeder as well as the silviculturist to have particularly the pest problem in control. And there are different strategies available. We also promote in the this institute has developed certain protocols where you can have mixed plantations. Even in the literature, I have seen Malina and teak can go together. We promote Casuarina and teak together so that short rotation, farmer gets the benefit of Casuarina and teak can go for longer time. Or certain countries like Brazil, they give a wider spacing between the trees, like 16 meter, 25 meter, like that. Something we need to promote depending upon the situation that should be decided. There is no hard and fast rule that you have to have this much only, something like that.
Okay. Right. Thank you very much. Yeah.
Thank you.
Okay, thank you. So before next section, so I would like to share information. 2 years ago, I went to Ghana during the first World Teak conference, and we asked the company how many clones do they have. So they told me that they have about 20 clones.
How many?
For the research. But the best clone that they use for the plantation only 4 to 5 clones that perform the growth performance very well. But the other, they do the research, try to make their hybrid between the clone that the best good growth performance, but they also would like to have more resilient to the climate change or other as well, as Dr. Yasodha and you mentioned. Okay. So may I take the next question either to Dr. Yasodha or Dr. Suwan, please? Anyone?
Dr. Ma, hand is up. Dr. Ma.
Dr. Ma, I'm sorry, I don't see your hand. Dr. Ma, please.
Hello, Dr. Ma.
Good afternoon. Can you hear me?
Yes, very well. Go ahead.
Thank you. Thank you very much, Dr. Yasodha, ICFRI in Coimbatore. And I learned so many things from your presentation. And particularly, I'm interested, I think the congratulations, the ICFRI selection of the superior teak material. And I'm interested also in India, the small farmer, the home garden, they are planting teak. So my question is, how do small farmer receive or access some high quality teak plantation? I learned from Thailand, also some of the private company, like the teak, what is the Thai, the Oki company. they are providing some the teak to farm while selling. In India, I think what kind of some mechanism while you're the institute? I mean, India is so large country, so how do you concern about the supply high quality teak material to small farmer? That is my question.
Yeah, thank you. So the question, how smallholder in India can access to high quality material, Dr. Yasodha?
Right. Dr. Ma, we have ICFRE has got a larger program currently going on for the past 5 years. In that, actually the institute had a long-term teak breeding program. From that, we had clones assembled. Around 45 clones we have in our clonal hedge garden. These clones we have taken into tissue culture route for propagation. And we are propagating on a large scale, and we are trying to supply to the farmers. Here, all other institutes of ICFRE which are working on teak, different parts, including Dehradun, one in Ranchi, Madhya Pradesh, Jabalpur, Rajasthan also included, and we all together we are working on the large scale production of teak. And actually, IFGTP has developed the protocol and given the clones to these institutes for large-scale production. And we also insisted them that locally they have to identify clones, because already plus trees were identified by the state forest departments. So they have to induct the clones from their own state, like that. For example, Madhya Pradesh, the institute has to induct at least 5 clones in culture. That they have done, and they are propagating on a large scale. Here, for large scale production also, we are not only depending on the government facility, we are trying to license out these genetic material to the private laboratories, private commercial laboratories. They are multiplying for us. Not only the farmers are getting the benefit, even the state forest department, because their requirement is quite huge, lakhs of seedlings they require. They prefer to buy the material from these licensee nurseries. Currently, this program is on.
Thank you very much.
Yeah,
Okay,
thank you very much. Thank you.
Just once again, and you also recommended some establishment of this coordinate teak gen pool. What I learned from previously in Malaysia, Sabah, or some Papua New Guinea, many countries they own they try to establish their own gene pool. So you have some recommendation how to coordinate, and what is some your suggestion what kind of some research coordination for teak from India, also other like Thailand and Malaysia. I think I like to learn from you some insight. And in addition, also I saw there's some in Sabah, what is YSG, the Dr. Dorian Ko is in the line. So I like also ask some idea about how to. My second question to Dr. Dorian Ko regarding the supply of the quality material, the small farmer, particularly the small farmers, they have very limited access, not like the large plantation, but I understand that Dr. Dorian Ko supply so many clones to large scale teak plantation, but I'm interested in learning in how to supply good material to farmer from Dr. Dorian Ko. But the question to Yasodha, the establishment, simple. I think some idea I like to learn from. Thank you very much.
Okay, thank you. So any thoughts from Dr. Yasodha, Dr. Suwan, and Dr. Dorian about the regional and international collaboration for sharing the genetic of teak?
Hi everybody. This is Dorian.
Hi, Dr. Dorian, go ahead.
Thank you for the question, Dr. Ma. we have never had any problem or issue with the distribution of clones from our company. We are, we welcome anyone who would like to access it. And so far, we have a lot of people coming to ask us to make use of our clones, and has been good. So yeah, in relation to a working collaboration, we are open to that, very much open. So if that should come up, we will be most willing to have a further discussion on how we can go about doing that. Yeah.
Okay, thank you very much, Dr. Dorian, for your commitment. So I would like to take the opportunity to announce that for the next webinar, Dr. Dorian will give a talk how to produce the good quality teak seedling. Okay.
Oh, okay.
Yeah. Okay. So
Thank
next
you for
question,
the short
please.
notice.
Yeah.
Okay. May I have a question? May I have a question for Dr.
Yes, please.
Yes. Dr. Yasodha, you mentioned something about the demand for the trends changing to young teak wood. How is that in the context for the Indian market? Would that be a big would that be a big interest in young teak wood for the market for the Indian market?
It is something about the marketing of the saying short rotation teak. Like actually, that India's importing all its teak requirements, and depending upon the diameter class and length, the market price is prevailing.
Yeah.
Of course, India has the market for different kinds of these kind of diameter class woods. According to the use, it gets the market price. I do not know whether I am answering right to you.
Yeah. Well, I'm just curious because I have known that the Indian market is quite huge in relation to teak timber. But then I'm surprised to hear that there may be a demand for young teak wood. As far as I know, I think it has always been older, mature teak wood being exported to India. So if in the event that there is a demand for young teak wood, I think that will be very interesting. And I'm sure a lot of people will be interested in that.
Yeah, that is mainly because of the pricing. I think it is like that. Of course, from Malaysia, we import many other timber also.
Okay.
Like Brazil and many of the South American and African countries import teak to India.
Yeah.
Okay, thank you.
Okay. Thank you.
Yeah, before I go to next question, I would like to provide information that for the global teak production each year is about 1.4 to 1.5 million cubic meter, and 94% of this go to India. So that's why Dr. Dorit asked that about the demand for the young teak wood in India. Okay. So next question, please.
Oh, can I ask a question?
Yes, please. Please identify yourself.
Yeah, I'm Muralidharan from Kerala. I used to work with the Kerala Forest Research Institute. My question is to Dr. Yasodha. You mentioned about genomic selection as well as use of CRISPR technology and genetic transformation. I was wondering, with the current status of research, how close are we to actually identifying the genes responsible for some of the important traits like straightness, or maybe even the hardwood color, which has great relevance for the price of the timber that comes into the market. So if you can have a quality planting material with these traits given some prominence, it would be a very important thing. So I would like to know from you, how close are we to actually identifying these on the basis of the molecular studies that has been done.
Thank you, sir. Genomic selection as a concept now we have implemented, and we are trying to analyze the hardwood content, hardwood color, as well as the metabolites in it. And we are trying to correlate. We know that the stem straightness or the color or the content are governed by multiple genes. The work is on, like a Brazilian group also, I was telling that they work on functional genomics. They are trying to figure out that what are the major genes influence these traits. And I don't think so that we are very near to come up with the genetically modified tree or something like that. I say that CRISPR technology or genetic modification technology need to be infused in teak also. Like to my knowledge, the short rotation trees like eucalyptus or poplars got multiple benefits through these technologies. Some of the private companies in Brazil, they depend only on the genetic modification technology for the production of herbicide tolerant or pest tolerant trees, particularly in eucalyptus. Likewise, when the need comes, when the most of the countries where teak is growing in as an exotic, they rely on clonal plantations only, and very limited clones are getting multiplied. So definitely possibilities of these kind of problems, pest problems are impending. Definitely we need to be working towards this, and we should have the alternate strategy to curtail such problems. That is what I'm trying to put in. In the current project on genomic selection, we are trying to establish the baseline. We need to develop the prediction models and other things. That is where we are into in. I don't think so. A project of 3-year-old or 4 or 5-year-old can give a solution immediately, but we get a lot of insights because of these things. In the next level only, we can go for the production as it is, production of quality planting stock or something like that.
Thank you, Dr. Yasodha. I have a follow-up question about the use of tissue culture for the clones in India in particular. I'm aware of the work done by Doreen and in Malaysia. But the impression I got was that the clones being multiplied and sold in India, most of the laboratories do not actually, are rather vague about the selection of the, they call it plus trees, but you hardly get any information beyond that. So I'm also glad that you have mentioned that ICFRE is taking the initiative of actually sharing the material with companies. I wish that was speeded up because the market otherwise will be flooded by material which is of uncertain origin. And ICFRE, I think, should take a lead in actually getting these best clones out into the market.
Yes, sir. Yes, sir. It is an all India coordinated project. The Ministry of Environment and Forest supported. So we are able to spread the technology across the tea growing regions in the country. Currently, around 11 or 10 different companies have taken the license of the material for large scale production.
Thank you, Dr. Yasodha.
Right, sir. Thank you, sir.
Thank you. Other question?
Yes, I have a general question for
Yes,
Dr.
go ahead,
Dr.
Dr. Noruddin.
Yeah, it's a general question. I know we are concentrating on teak, and of course, we know the importance of teak. And with teak being the highly traded tree crop globally, there's always some money to be dedicated to research. I'm wondering whether we have maybe similar genetic study in terms of tree breeding for other tree species from your lab, Dr. Yasodha, and then that of Dr. Swan, because we cannot over-concentrate on one tree species because we have to look at other tree species so that we can, at the same time, continue to have a balance in terms of what we have in the market for trade. And we also need improved, genotype to be able to also have other tree species that do not grow fast, especially from the Africa region. There are a lot of tree species out there, very good, high quality, but they don't grow fast. And of course, tree breeding definitely will be, will be the answer to that. Do you do other type of research looking at other tree species other than teak?
Yeah, this question is addressed to me? Similar to teak, already this institute is Institute of Forest Genetics and Tree Breeding with a specific mandate of generating improved planting stock for the identified trees. So this institute was established somewhere in 1988 by the ministry. We have the national mandate for the whole country. This institute has to develop the do the breeding. So accordingly, we have taken up currently around 12 species are in the higher levels of improvement. Specifically in the case of eucalyptus and casuarina, we have used the breeding tools, and the clones are growing throughout the country, I can say. And in the productivity is more than doubled compared to the seedlings. That is the efficiency of clonal production. Then other than that, we work on Nulamarkia, Kadamba, Malina, Tamarind, Dalbergia, like that. More than a dozen species are into it. Nowadays, for the past 5 years, the council, the ICFRE, has taken this as a all India coordinated mode, all India coordinated research project like that. So for 5 years, we are trying to execute the tree breeding plans involving other institutes. Not only the ICFR institutes, other institutes are also involved. Even the state forest departments are into it. So the work is in progress.
Thank you very much. What about Thailand, Dr. Swan, and maybe other areas?
Other economic species, Dr. Swan, in Thailand, besides teak. You mute microphone, Dr. Swan.
Oh. In case of Thailand, we have eucalyptus and acacia, but I think when we think about this kind of question, we have to think about the market, right? Of course, we have, I mentioned eucalyptus and acacia, but these two species, different use, different utilization, or even, I think, different market. For teak, I think there is no question about the wood market. That's this species is in international market already. So I would say that it's very difficult for Thailand to make more species in terms of the native species. We have put a very big effort for teak. So far, maybe I would say almost 60 years already. But what I think when we think about the tree improvement program, there are several aspects that we have to think. I already mentioned the market and also the resources that we have. For teak, we have very good resources, and also in terms of the natural distribution, this is wide enough. We have a very big genetic base from India, Myanmar, Thailand, Laos. So with this kind of wide distribution, if we develop the tree improvement program, it would be more effective. So I would bring you to the question that we already we mentioned about the start of the regional cooperation project. I will focus again that making the coordination of the gene pool establishment very important. And we know that provenance test or provenance effect much bigger than individual. It means that if we, for the good start of any species, we have to get start from the progeny test first and see which progeny is good for which site, which country. So if you ask me about the regional cooperation project, it would be something like international cooperation on the gene pool correction of this species. And I think each country will get the benefit from this kind of project. And I think I would like to say hi to Dr. Doreen Go, no? So very great to meet you again. Very impressive. In the past that I learned the mass production of your tissue culture export to several parts of the world. This is my point. Thank you.
Thank you.
Thank you. Thank you.
Other question from the floor?
Yes, I have one question to
Yes,
Dr.
Dr. Turasidas, please. I
Yeah,
recognize your voice.
This is related to Suvan's presentation with his comment on the correlation of hardwood proportion with the diameter of the teak tree. He says it is of poor correlation with the diameter. But how is it possible? It is proven that if the diameter of the teak, the short timber is short rotation teak, is higher, the proportion of hardwood is also naturally higher. But because the short rotation timber production is mainly aimed at accelerating the growth by silvicultural activities, by performing silviculture thinning and other activities that promote tree growth in diameter, that actually promote higher proportion of hardwood. So now Dr. Swan says that it has got poor correlation with the diameter. How it occurs? I have some doubt with regard to that, because all over the world, the short rotation teak planters or the forest management is aiming at accelerating the tree growth by giving fertilizer application, then performing the periodic thinning and silvicultural activities to maximize the production or the productivity from plantations. So if the hardwood proportion is not related to diameter, how this short rotation plantation activity will, whether it will be adversely affected by this poor correlation you mentioned. Yes, Suvan, Dr. Suvan, please.
Did you hear the question, Dr. Suvan?
อาจารย์ช่วยสรุปให้ทีถามว่า
Okay. So the question is from your presentation, you show that there is a low correlation between the height and the diameter and of the hardwood. This may be due to we enhance the silviculture technique and fertilizer in order to maximize the growth from the short plantation. So if we have the longer rotation, let's say 50 year or 30 year or 50 year, so is the correlation will be different from your presentation?
Well, we actually at the moment we have no clear evidence yet, but it seems to be that we believe in that way, no, longer time and could be longer high proportion of the hardwood, but still I would say no scientific evidence from now. Yes.
So it's only your observation, no scientific.
Yes.
Okay.
And I have one more question to Dr. Yasodha with regard to her mentioning in her concluding part that the selection, the breeding or the genomics, which can lead to higher proportion of selection of trees having higher hardwood proportion, then increased extractive content of material, no? So how big this extractive content is related to the heritability? It is not related to the heritability, I would say, because this is more related to the age of the tree. The polyphenolic content increases, or the extractive content increases with the age of the tree. Not we can induce by selection of any genomic selection possible with regard to inducing higher polyphenolic compounds in teak, which will ultimately increase the durability of the timber. But that is not possible in the short time by inducing extractive content through genomic selection. This Dr. Yasodha, this question to her to answer, please.
Right, sir. Right, sir. Like I listen to you. Like there is a high amount of variability existing in the hardwood content.
So Yasodha, can you talk close to microphone, please?
Yeah. Like the secondary metabolite content, his variability is very high. When we say genetic variability in teak is very high, even the secondary metabolite content is varying across the genotypes. So currently we are working with around 140 clones, and we could see certain differences using the FTIR spectroscopy. There is a variation in the secondary metabolite content. So by using the genomic selection tool, we can select the individuals which has the higher secondary metabolite content as well as the higher level of hardwood content.
Of the same age tree. Trees of
Yes,
the
sir.
same age.
We are actually analyzing different age classes from 9 year onwards. Different plantations. Suppose all these things have been done with the help of Kerala Forest Department. Different age classes, we have taken the samples and we are working on it.
Okay, thank you. Can I go to the last question to Dr. Ma? Yes, go ahead, Dr. Ma.
Not question to speaker, but the recommendation to Professor Yongyu yourself. So today we had a wonderful discussion regarding the next webinar that you announced, Dr. Dorenko, and I'm very happy. And perhaps you may consider also inviting the expert from the teak farm in Cambodia. I think that they are really advanced, innovative approach using irrigation and fertilizer. I remember they are really advanced, like somehow 7 or 8 year the harvesting rotation. While most of
excuse me, can you come closer to the mic, Dr. Ma? Sorry.
Can you speak louder, Dr. Ma?
Sorry. Now is it better?
Yeah.
Oh, thank you. So just question, not question to speaker, just recommendation to Professor Yongyu yourself. Regarding next topic of the webinar, and I think already you also catch some innovative teak plantation in teak farm in Cambodia, where they are looking for harvesting about 7 or 8 years. As I understand, most of the teak plantation from clone teak, of course, they are working for like 18, 20 years. So it will be very good to invite first the teak farm in Cambodia, along with Dr. Dorenko in Sabah, so that next webinar can provide a wonderful opportunity to really the progress of the clone teak plantation in innovative way in Cambodia and other parts of the tropical country. Thank you.
Okay, thank you, Dr. Ma, for your recommendation. In fact, Dr. Dorian already committed to give a talk in next webinar. So we will contact her after this one. So on behalf of the organizing webinar, I would like to thank both speakers very much for your excellent presentation. Okay. So before we close the session, I would like to summarize from the today webinar. So we learned from Dr. Suwan that the natural teak plantation, the natural teak forest, right now cover in 4 countries: India, Myanmar, Thailand, and Laos. Is about 29 to 30 million hectares. So in Thailand, most of them are located in protected area. And beside that, Thailand, we conducted the ex situ conservation and research in 3 eras, starting from botanical gardens, seed orchard to codon test and DNA. However, based on his observation, so he found that there is less correlation between the height and the diameter of the hardwood. But however, Dr. Yasodha from India, based on the genomic innovation and research, so she indicated that the genomic technology can enhance the quantity of hardwood by using the technology. However, this is the trade-off between the genetic diversity and the growth performance. Because if we concentrate on the hardwood only, we will select only a few genomic diversity, and we may lose the genetic diversity. That one will have the consequence on the resilience of the teak to the climate change or other environmental impact in the future. And both speakers have the what you call the same recommendation, that the regional cooperation is very important to share the knowledge and to share the genetic resources to enhance the what you call the good quality planting material. So once again, I would like to thank both speaker and the comment from the floor. So before we go to the closing session, I would like to invite Dr. Turasidas to provide information about the 5th World Teak Congress in Kerala in September. Dr. Turasidas, please.
Yeah. This a couple of days before, we had a meeting with the ITTO-Teak team with regard to the side event which is going to be organized on the opening day itself on 17th September 2025 afternoon, maybe in the evening around, for a 2-hour session. It is proposed from 4 to 6 p.m. in the evening, in which it was reported by Dr. Tetra that the executive director of ITTO will give an opening remarks, and 6 presentations from each country is anticipated. It is, I think, yesterday, Professor Yongyut circulated the tentative participants list to the ITTO team to nominate people for participating in the World Teak Conference. In the meantime, because it is 2-hour session with 6 presentations are anticipated from each country. And in the meantime, if anybody has submitted abstract for presentation in the WTC before the deadline, whether they received the confirmation for oral presentation, or if they want to change their presentation to ITTO session, it is convenient to take up that case, because in that case, just like I heard from Professor Yongyut told in the meeting that Tunai Institute already submitted abstract for presentation. and also Dr. Yasodha from IFGTB. So if those two presentations are shifted from the already submitted sessions, it will be convenient for ITTO session not to repeat again requesting abstract from the participating countries. So we need not duplicate the work. So if they are interested, that can be shifted to the ITTO session. So that is one thing. And if they want to retain in the respective sessions, already they submit the abstract, and if it is accepted, that's also good. It is up to the presenter if they would like to shift their presentation to the ITTO's special session. So that will also give an opportunity for the other waiting presenter to be considered for oral presentation, because only limited 3 presentations for each session is allowed. So this is what I would like to share with the forum now.
So thank you very much, Dr. Turasidas, for your informative information about the Fifth World Teak Congress in Kerala, India, during 17 to 20 September, and post the excursion on 21st and 22nd of September. For the ITTO and BMLEH Teak project, we will organize a side event on 17 September. It means the first day before the reception party by the organizing committee. So we encourage everyone who are interested to apply and register. So the deadline for the early bird registration is on 30 April. But for the participating country, we will request the permission for the group registration with the early bird registration fee, only $200 per person. Thank you very much, and we will share with you for further information about this. Okay. So may I invite Dr. Tetra, the ITTO project manager, to give a closing remark on behalf of ITTO. Dr. Tetra, please.
Thank you, Professor Yongyut, for your fait accompli to ask me to close this webinar. Yeah, I was enjoying the discussion, and thanks, of course, to the resource person, Dr. Yasodha, and Dr. Suwant for very excellent presentation, and also for the participants, all participants, and especially for the participant that put forward question to enrich the discussion in this late afternoon webinar. So I hope that hot ideas will be taken as an idea to move forward to strengthen the collaboration and networking among us. So we are really looking forward to have more concrete and robust partnership among us, so that we can talk more about the TICs, future of TICs, and what we can do more to give value added on the TIC and TIC products. So I hope that you all enjoy the webinar, and hope to see you again with more participants in the next webinar, which probably will be organized a month from now. So thank you so much. Thank you so much, and have a good evening. Thank you.
Thank you.